Today, I'm going to be talking about genome editing, also called gene editing. So, gene editing is a group of technologies that gave scientists the ability to change an organism's DNA. These technologies allow genetic material to be added, removed, or altered at a particular location in the genome. Several approaches to genome editing have been developed, and a well-known case that we're going to be talking about today is CRISPR-Cas9, which is short for clustered regularly interspaced short palindromic repeats and CRISPR-associated protein 9. The CRISPR-Cas9 system has generated a lot of excitement in the scientific community because it is faster, cheaper, more accurate, and more efficient than other genome editing methods. The CRISPR-Cas9 was adapted from a naturally occurring genome editing system that bacteria use as a natural immune defense system. When infected with viruses, bacteria capture a small piece of the virus's DNA and insert them into their own DNA in a particular pattern to create segments known as CRISPR arrays. And the CRISPR arrays allow the bacteria to remember the viruses or closely related ones, so that if the virus attacks again, the bacteria will produce RNA segments from the CRISPR arrays that recognize and attach to specific regions of the virus's DNA. And then then the bacteria then use the Cas9 or a particular enzyme to cut the DNA apart, which disables the virus. So researchers adapted this immune defense system to edit DNA. They'll create a small piece of RNA with a short guide sequence that attaches or binds it to a specific target sequence in the cell's DNA, much like the RNA segments that bacteria produce from the CRISPR arrays. And this guide RNA also attaches to the Cas9 enzyme. And when introduced into cells, the guide RNA recognizes the intended DNA sequence, and the Cas9 enzyme cuts the DNA at the targeted location, which mirrors the process in the bacteria. So although Cas9 is the enzyme that is used most often, other enzymes like I believe CPF1 could also be used. But once the DNA is cut, researchers will use the cell's own DNA repair machinery to add or delete pieces of DNA material or to make changes to the DNA by replacing an existing segment with a customized DNA sequence. Genome editing is of very great interest in the prevention and treatment of human diseases. And currently, genome editing is used in cells and in animal models and research labs to understand diseases. And scientists are actually working to determine whether this approach is still safe or is safe and effective for use in people. And it is being explored in research and clinical trials for a wide variety of diseases, including single gene disorders such as I think it was, I believe, cystic fibrosis, hemophilia, and sickle cell disease, if I wasn't mistaken. And it also holds promise for the treatment and prevention of more complex diseases like heart disease, cancer, some mental illnesses, and I think it was human immunodeficiency virus, HIV. Um, but some ethical concerns also do arise when talking about genome editing because using technology such as CRISPR-Cas9 is used to alter human genomes. And most of the changes introduced with genome editing are limited to somatic cells, which are cells other than egg and sperm cells, germline cells. And these changes are isolated to only certain tissues and are not passed on from one generation to the next, as we know. However, changes made to genes in egg or sperm cells or to the genes of an embryo could be passed to future generation. And so germline cell and embryo genome editing brings up some ethical challenges, including whether it could be permissible to use this technology to enhance normal human traits such as height or possibly intelligence. But based on concerns about ethics and safety, germline cell and embryo genome editing are currently illegal in the United States and many other countries. And on top of that, it is pretty costly. But yeah, that's my short video on CRISPR-Cas9 editing. Hope you enjoyed.